All right. Hi, everyone. Welcome back to our get-together on the cerebellum. And today we have Clement Lena, who I'm so pleased to have with us. Clement started his scientific career as a quantum physicist, and he found it much more interesting to study neurobiology while he was at Ecole Normale Supérieure in Paris. Then he moved to the Marie Curie University, also in France, where he did a PhD in neuroscience, studying the interactions between nicotine, dopamine, the neurobiology of addiction. Then he came to the United States, did a postdoc with Matt Wilson at MIT, and then returned to France to head a group at Ecole Normale, along with Daniela Popa, where he's been studying the interactions between the cerebellum and other structures in learning, particularly things like fear conditioning. Clement, such a pleasure to have you. Thank you for your time.
Thank you, Reza. I must say I'm extremely grateful for this seminar series, which is absolutely fantastic, and thank you infinitely for doing this. And I must say I'm incredibly honored to be invited to present our work. So thank you very much for this invitation. That's something for me. Okay, so as I was saying, the central topic for our team is to try to understand what's happening in the cerebellum-forebrain coupling from the neurophysiology side. And what I'm going to show you is some recent work from our team in Paris on two systems, the sensorimotor and emotional systems. And these work for this presentation is aimed at illustrating the complexity and the functional diversity of this cerebellum-forebrain coupling, which I think is far more, the more it goes, the more we realize how complicated it's going to be to figure it out and to understand all the variants of this coupling and what it's used for. Okay, so I'll start just with very, very basic things. I think everyone in the audience is very much aware that despite its small size, the cerebellum is packed with neurons, that the proportion of neurons found in the cerebellum and the cerebral cortex actually has been pretty stable across the mammalian brain evolution, as you can see here. So there are details depending on species and primates. Humans are slightly bigger cerebellum than would be expected, but still, I think generally there's a very good indication that some evolutionary pressure is applied to this system. And the common belief, because in evolution it's never easy to demonstrate anything, is that the reason for this coevolution is to be found in the fact that the cerebellum and the forebrain, particularly the cortex, have these reciprocal connections. So I think, as most people know, these connections are not direct, neither way. Pretty much the whole cortex projects to the cerebellum by way of the pontine nuclei, and the cerebellum projects back toward the forebrain, the cortex, notably through the thalamus. So this is kind of textbook description. Actually, now, if we really want to be on page, I think we should, of course, include the vestibular and parabrachial nuclei, which are similar to the cerebellar nuclei in terms of position in the circuit. And of course, we should not forget about the midbrain as a relay between the cerebellum, of the action between the cerebellum and the forebrain. And last but not least, of course, the basal ganglia have joined the structure that form these complex reciprocal interactions. So what's the cerebellum is good for is it's definitely doing something in the motor domain. We know that for more than a century. And it does that with a very simple circuit, which has been very well characterized. And of course, I'm not going to detail here, but that strikingly seems to be quite uniform, although not perfectly uniform. And the more it goes, the less uniform it is. But still, it's the same sort of circuit that you can find all over the cortex and the nuclei underneath. So this circuit is used for motor function, and we can see here, as an example, the recruitment of the cerebellar cortex in motor on somatosensory processing. in the human cerebellum on these maps. And as everyone knows, there's plenty of gray area here, and that's because these parts of the cerebellum are far less involved into sensorimotor processing and a lot more into other functions related to cognition. So what the cerebellum functionally does is far more understood in the case of the motor system. And just to try to frame it, and of course, this is an oversimplification, but what is pretty clear is that the cerebellum is very good at improving movement accuracy by learning to anticipate short-term deviations from the plan. So whenever things are going to go wrong, actually the cerebellum has the ability to anticipate that even before the feedback of the sensory system, which is taking some time, has taken place, and help the brain to figure out what comes next and how to correct any deviation if needed, or take any other actions, right? So on the cognitive side, I must say, for me as a neurophysiologist, things are pretty clear in terms of there's something, and pretty fuzzy in terms of what is it exactly. I think we know now for more than 30 years that it's pretty well established, notably helped to the work, of course, of Jeremy Schrammman, that the cerebellum contributes to cognitive function. We know that lesions in the cerebellum can lead to the famous cerebellum cognitive affective syndrome, which is a complex syndrome that has many aspects and many forms. But how to connect these things and how the circuit is operating on the same thing is not so clear. So very early on, Masao Ito had pushed up this idea that we could transfer the understanding from the motor system to the cognitive or other aspects of cerebellar function. And you can see that 20 years later, this fantastic read, this consensus paper from so many leaders in the field was still, pushing this idea that somehow the understanding in the cognitive, what's happening in the cognitive domain stems from the understanding of what's happening in the motor domain. So this is how it would work. So the idea is on the uniform circuit of the cerebellar system implements always the same sort of computation. It's a form of supervised learning with so anti-Hebbian and recent past input. Of course, again, this is an oversimplification. Things are getting more and more complicated the more we know about it. But still, the idea is that the cerebellum has this ability to perform association between events shortly in times, between events and results of the event shortly in times. And in the motor domain, it's pretty clear that what the cerebellum does is something that can be described as implementing internal models. Briefly, that's all about making short-term state prediction and using these state predictions to process motor commands and somatosensory feedback in order to tune up things, right? And at the end, the common idea that has dominated also by lack of something else, of course, is that you can then decline this sort of function in various computational tasks, which can be motor control, but emotions, cognition, and so forth. And I'm putting this figure because I find it's a very inspiring work from Didriksen and colleagues. I think Richard Ivry has shown that also in his in this very seminar. Actually, I think the idea that we don't need to really discuss or try to understand what's happening in the algorithmic level is really coming into questions now. And maybe it becomes interesting to try to think this problem of understanding what's happening there by opening up the sort of actions that the cerebellum is doing in the brain. Okay, so that's typically this intuition that we've been following now for more than 10 years in the lab here. We work in rodents. The reason for this is that, well, they have a cerebellum, that's mammalian brain, and we have all the benefit of the modern tools for high-density neurophysiology, but also for optogenetic, chemogenetic, and many other approaches that are very handy in rodents. And so what I show in two parts of this talk, I'll discuss stuff in the somatic sensory motor system, and then the other part will be in the emotional system. Okay, so the system that we've studied most in the lab is the whisker system of the rodents. So the whiskers are these long facial hairs that you find in the rodents, and particularly nocturnal rodents. They are mobile, and they're essential for moving around and figuring what's in the environment, because of course the animal live in the dark. So if we look in the somatosensory or motor cortex, in fact there are very substantial portions of the cortices that are devoted to that system, despite the fact that they occupy only a small fraction of the body surface, right? It's almost as, in the primate human hand representation, except, of course, the rats and mice cannot grab anything with their whiskers, of course. So these whiskers are moving. In fact, there are several sorts of movement, but the most prominent one is an oscillatory movement called whisking that allows the animal to perform something equivalent to palpation of the environment in order to figure and recognize what's around. Okay, so if we look in the literature, we know that these whiskers, whenever an animal is moving around and exploring what's happening, they're moving, and they're in fact adapting their position very finely in order to study what's happening. So here you can see, I hope if the resolution is not too low, that when the rat approaches an object, a new object, it's progressively adapting its movement, and you can see on the right that the oscillation becomes progressively asymmetric, and in order to so with a more advanced protraction on the side that needs protraction to get into contact with the object. So very fine tuning. Well, that was enough for us, because if there's fine tuning of movement, there must be cerebellum, and it's worth looking into it. So that's pretty much all stuff, but I want to show this because I think it's the rest is not to say that the main way of the interactions, or the most classical way of interactions between cerebellum and cortex is what I'm going to show afterwards. We recognize fully this mode. So what we did in the past was perform some metabolic imaging to identify the part of the cerebellar cortex which we're receiving from the motor cortex. We figured that, in fact, the same region of the cerebellar cortex was receiving from the somatosensory cortex as well. And even more than that, single cells in the cerebellar cortex were receiving both from the motor and somatosensory cortex. So what the processing involves some convergence. And the group of Daniela Popa has since then really extended this view that, it's not a point-to-point, but there are definitely convergences taking place in that system. On the other hand, to map the other way, we use optogenetics. We found that the region that triggered some LFP signal in the motor cortex most efficiently was located exactly at the same spot that the region that was excited by the motor cortex. So definitely we had some closed loops. And then what was happening in this closed loop in terms of movement control could be studied by just measuring whiskers movement. When the animal starts whisking, some activity develops in this loop. And if we perform optogenetic stimulation of Purkinje cells, which are inhibitory, then we can break the loop. We can stop the activity in there. And when we do that, we see that it does not interrupt the movement. The movement does not require the cerebellum, but it definitely shifts its parameter. And selectively, what we found is that it's shifting the set point of the movement, the set point of the oscillation. Since then, other groups, notably the one of Paul Shadmehr, has shown that Purkinje cells indeed encode this parameter. But at that time, it was still unclear whether the impact of our stimulation was linked to the pathway linking the cerebellum back to the cortex, or whether it was projecting elsewhere. So to look if it was the case, what we did is we blocked the motor thalamus. We inhibited the motor thalamus and performed the same inhibition. And in that case, we found that there was no effect on the set point anymore. So it was really the interruption of the loop that was shifting the motor system. So basically, what this study showed by point-to-point and physiological and anatomical tracing is that we have loops that's very consistent with things that have been looked at since the 70s or 60s. And the neurons that are projecting toward the effectors make those collaterals in the pontine nuclei, which are related to the cerebellum, and then go back to the cortex. So there's a feedback that goes through the motor thalamus. And these motor thalamus typically belong to one specific class of thalamocortical system. You'll see why I'm talking about these classes, called monofocal thalamocortical system by this recent work of Francisco Clasca. So what we found, and was a little less classical compared to the literature, was that this feedback from cerebellum to the cortex was in fact enriched from somatosensory information that was coming both from the peripheral system in a short way or through the somatosensory cortex, and was working in there. Okay, and this circuit has exactly all the things that are needed to, plus we know the computational properties of the cerebellar cortex. This circuit has everything that is needed to improve motor control despite delays in the sensory processing, which is the standard model of cerebellar interactions. Okay, so overall, this is just for whiskers, but we know that mammals have hundreds and hundreds of muscles. So we expect, in fact, that the cerebellocortical system is, in fact, containing many of these loops, or extended loops if we add the somatosensory cortex. And that's definitely going to take a fraction of the functional connectivity between the cerebellum and the cortex. Yet, that's not necessarily the whole story. And the first thing that came to our attention was this experiment that we did with Daniela Popa some years ago, where we recorded from the somatosensory and motor cortex at the same time in rodents, so while they were performing some free exploratory whisking in their environment. And what we observed is that during whisking, there was some coherent activity that developed between the motor and the somatosensory cortex, the primary motor and primary somatosensory cortex of the whiskers. And this was particularly taking place between the deep layer of the somatosensory cortex and the superficial layer of the motor cortex, which turn out to be the ones that have the densest reciprocal connections. So what are these coordinated activity? Here you can see the coherent spectrum, which is identifying for different frequencies, or you can, if you don't believe in brain oscillations, you can look at it as time scales, where there seem to be systematic coordination of events in the two structures. And as you can see, there are multiple peaks in there that seem to signal that there are different time scale or frequencies over which the activity between the two structures is taking place. So what captured our attention particularly were these gamma band oscillations. So that's above 25 or 30 hertz, which is faster than the whisking frequency in the rat. And gamma oscillations are interesting because they're a very good sign. They're observed when sensory processing takes place. They are linked to signal propagation by many sorts of arguments, which I'm not going to detail. So gamma oscillations were the sign of something that had to do with sensory processing. So that was it, right? So we recorded from a bunch of rats and performed. So here you see the average spectra of this. And what we did then was inhibit the cerebellar nuclei by a local injection of a GABA agonist. And we found that this was not because of the doses and place. This was not such an impairment for the rats. The rats were still moving around and whisking. The thing that was most prominent was that when we looked at the coherent spectra, the gamma coordination was gone. And if we look at the difference between these two curves, so the control in black and the cerebellar inhibited in red, so that's the curve on the bottom left here, you can see that there's a very clear, so these are pairwise difference from each rat. You can see there's a clear depression of gamma coordination between these two structures. I must confess that when we found that, we were kind of worried because it was absolutely non-standard. So we verified a number of things. It was not because gamma oscillations were gone in the two structure. Gamma was still there. Local cells were still phase locked to the local gamma. So that was not the simple explanation. We couldn't understand that. And that was kind of a relief to finally find that recently Chris Deseo's team managed to reproduce in a slightly different setting the same observation in mice, contrary to our rats. But as you can see, they also find the same change in coherence following cerebellar inhibition. Well, that's a new idea. The cerebellum can regulate the coordination of activity in distinct cortical structures. And the recent work from the lab of Detlef Heck the anterior part of the brain is completely following the same idea and receiving further experimental support. So there seemed to be something into that. So how could that work? That has been really an lingering question for us. We felt that the initial circuit that I just showed you earlier, where the cerebellum feeds back through the motor thalamus essentially to the motor cortex, it was very hard to find a way, not impossible, but very hard to find a way, convincing way, where we would lose the gamma coordination by removing the cerebellar input to the motor thalamus. Instead, we came up with this idea that maybe there was some relay between the cerebellum and the cortex that would have the ability to coordinate, in fact, what was happening in the somatosensory and motor cortex. So the big question was, what was this relay? At that time, there was very little evidence that the cerebellum was projecting to the somatosensory cortex. What we did then was to look for it. We knew that there was no direct projection from cerebellum to cortex, right? So we had some cholera toxin injections in the somatosensory motor cortex to retrogradely label the neurons that were projecting to each of these structures. We injected M-cherry in the cerebellar nuclear neurons to visualize the nerve terminals. And that was it. We just sliced the brain and looked for places where there would be cerebellar terminals and neurons projecting to the primary somatosensory cortex in green here, or motor cortex, or both in yellow, right? And that's what we found. We found that the majority of the neurons that were putatively receiving from the cerebellum and projecting to the somatosensory cortex were found in the posterior medial thalamus. It's one of the thalamic nuclei, but we also found in more classical cerebellar targets, the ventrolateral and ventromedial thalamus. Interestingly, we found that there were a bunch of neurons that were projecting both to the primary somatosensory cortex and motor cortex. And particularly, we found them frequently in the posterior medial thalamus. We also found a few of them in the other structures. So these captured our attention because the posterior medial thalamic neurons are belonging to another type than the ventrolateral thalamocortical neurons that Francisco Klaska called the multifocal thalamocortical neurons. And these neurons have these special axons that are branching out and making dense projections into distant groups of regions. But it's not widespread, it's targeted, but it's really allowing to send some information simultaneously in multiple structures. Okay, so where the POM neurons really receiving from the cerebellum? So we looked at this with some friends in Paris, where we expressed the channel rhodopsin in the cerebellar terminals, performed some slice, and found that in slices, the optogenetic activation of cerebellar terminals was, in fact, potentially exciting the neurons. Here you can see a big burst produced by the cerebellum-POM pathway, and very nice depolarization. And these stimulations could still produce very substantial currents even if we repeated them at a relatively high frequency. So there were not a massive adaptation, which would prevent some continuous transmission. So this is the signature that the cerebellum-POM pathway is very efficient. Okay, but are these POM neurons projecting to the somatosensory cortex? To look at it, we looked a little bit more into detail. So the competition possibly could be with the ventrolateral thalamus, which is not far from the posterior medial thalamus. But we know from this classical paper from Kuamoto and colleagues that the ventrolateral thalamus neurons that receive from the cerebellum actually do project to the intermediate layers of the cortex, and particularly they don't project to the layer 1 of the cortex. It's only the neurons that receive from the cerebellum, from the basal ganglia, sorry, that are projecting to the layer 1 of the cortex. And on the other hand, actually the posterior medial neurons have this ability to make, so you can see a single axon here, which is branching in the somatosensory and motor cortex region. And in that case, they branch, of course, in deep layers, but they also branch quite clearly in the superficial layer, in layer 1. So what we did then was to record from the layer 1 boutons stained by injections of GCAMP in the posterior medial neurons, and performed optogenetic stimulation of the cerebellar nuclei. So when we do that, here are just a few boutons that we isolated from our 2-photon experiments. You can see that every now and then you find some boutons that are activated by cerebellar stimulations. Here you see very strong calcium increase. And if we look over time, it's something that is very well maintained. So it's really consistent with the fact that in the somatosensory cortex, the cerebellum can produce short latency activations of axons that are reaching the layer 1. So we can do that on a bunch of terminals, and you can see that the average response here is really going very, very fast following the onset of the stimulation. So layer 1 is filled with axons that are coming from all over the place and may not be actually functionally related to whisker processing. So we felt it was a good idea, in fact, to check if there was any functional overlap between the POM axons that were excited by the cerebellum and the ones that would be excited by whisker stimulation. And actually, we found that a number of these terminals are responding to both sorts of stimulation. So we are dealing with a somatosensory related posterior medial neurons that receive with the cerebellar inputs. Okay, so are these able to coordinate the activity into structures? To look at this, again, we started recording from the somatosensory and motor cortex at the same time. We recorded the local field potential in the superficial and deep layers. And as you can see here in this nice whisking episode, there are the oscillations in the somatosensory cortex in blue and motor cortex in red are quite nicely observed in the two structures. You can see that the amplitude is not identical. The fluctuations of amplitude are not identical. The difference is not exactly 0, but there's definitely some coherent activity. And if we compute the coherent spectrum only during whisking, you can see that there are definitely coordinated activities that are taking place at various timescales. So now what happens if we disrupt the cerebellum-POM pathway? So we did that with the intersectional approach with viral injections in the POM and in the cerebellar nuclei. And then when we added some CNO, so when we inhibited those cerebellum-POM neurons, we found that there was a disruption of the coherent activity between the two structures. There was no change in the power, in the gamma power. And we found that, in fact, the pairwise difference between these curves was indeed showing some significant loss of coherent activity between the two structures. Okay, so from the initial loops that I was showing, which is the classical cerebello-cerebral coupling, what we found is that, in fact, the cerebellum can interact with multiple brain regions at the same time via a different pathway. Even if it's the same motor cortex, in fact, there's one interaction through the valve, and which goes to the corticopontine nuclei. And there's another one which is, in fact, known to target the intrateleoencephalic neurons. So these posterior medial axons don't have powerful synapse on the corticopontine, but much more prone to contacting these intracortical intrateleoencephalic neurons. And therefore, this allows these thalamic neurons and the cerebellothalamic neurons to gate the intercortical communications. So this is made possible because it's operating through multifocal thalamic neurons. So these multifocal thalamic neurons are not only found in the posterior part of the thalamus, and here you see a little list of multiple thalamic nuclei that host these sort of neurons. You can see the pulvinar, which has incredibly expanded in the primate brain, lateral dorsal, medial dorsal, which is the prefrontal area, which projects to the prefrontal area, medial geniculate, and that's it. So it turns out that the cerebellum is contacting many of these nuclei. There not that much evidence in the anterior group. Maybe that will come. But it seems that this mechanism that we describe, this mode of operation of the cerebellum, the cortex, might well generalize to many other systems and many other functional circumstances. Okay, so to wrap things up, before wrapping it up, sorry, I'm going to shift gear and move to the emotional system. So the idea that the cerebellum is involved in emotion is not new. We know that for a number of years, but there are two sides of this. In emotions, there is a lot going on about the expression of emotion, which is a sort of motor function. But the other side of emotions, which we felt was very appealing, is about emotional learning. And this is interesting because we know by experience for a human, we know that a lot of our emotions are not coming simply because we see something horrible or we see something joyous, but it's also because we've associated things in the past circumstances with emotions, and coming close to the circumstances is enough to trigger emotion. So there's a lot going on the memory side in the control of emotion. It turns out that the cerebellum is very much linked to emotional memory, and you can see in this study from Damasio and colleagues that the recall of emotional memory in a set of subjects is actually powerfully activating the cerebellum. I must say, what I find slightly frightening is that it's mostly the negative emotion that seem to require the cerebellum. You don't need much of a cerebellum, apparently, if you're happy. So what could that be doing, right? Particularly, it's pretty clear that most of the emotional learning, I'm not talking about eye-blink conditioning, most of the emotional learning are not cerebellar, right? So we started with a very non-cerebellar learning, which is the cued fear conditioning. I guess everyone is very familiar to this. So you have a box, you put mice in this box, and then you start playing a sound, and at the end of the sound, you produce an electrical shock. So mice don't like it. They start showing some trace of caution, feeling of threat, and this is shown by immobility, which climbs up. So of course, at the end of the session, we don't necessarily know whether the mice have associated the sound with the fear. They just feel some threat and express some behavior, but we know that there's memory because the next day, if we go to different contexts, we don't play sound, the mice are very comfortable, don't showing threat-related behavior, but the moment we play the sound, there's a big jump and the mice start showing it. If you use cued fear conditioning, it's interesting because you can study fear learning, but there's another learning that you can study at the same time, is the fear extinction learning. Fear extinction, the thing that happens if you play the cue, the sound many, many times. So we do it 25 times a day over 3 days. And if you do that progressively, you will see a reduction of the reaction to a threat, where the animal go back to something that seems normal life. So it would sound that the animal just forgot about the association between the sound and the threat, but that's not the case. And a huge literature over several decades now has shown that it's a separate sort of learning. And what the animal learn is to suppress this threat behavior. And in fact, if we look at the brain structure responsible for that, of course, it's not brain structure, it's a circuit, but the key structure for fear learning, for acute fear learning, is the amygdala. And the key structure for the fear extinction learning is the medial prefrontal cortex. If I want to complete the schematic for the purpose of this talk, I should add the periapical gray region, which is a central structure for defensive behavior. And this structure is playing notably a role into the expression of the fear response, or the behavioral response to threat. Okay, so except for some work from the group of Strata earlier on, there's not so much evidence that explains how the cerebellum could be playing a role in that circuit. There are indications, there's potential connections, but for a very long time, we had very little experimental knowledge of what could be happening in there. So one thing that is still interesting that has to do with prediction, cerebellum is very much into prediction, is the Rescola-Wagner. So the Restler-Wagner learning rules, from the name of these authors, is something that applies to some forms of associative learning between cues or context and salient events, either good or bad. And the key idea of this learning rule is that there is a certain degree of association between cues or context and salient events, or something bad or something good can happen in there. And every time there's a new exposure to this association, there's an update of the memory that is taken, or the memory of this association in the system. And this update is not simply the reflection of the event, but in fact, this update, the size of this update is proportional to the unexpectedness of the event. That is, if in a given cue or context, in presence of a given cue or context, this salient event is entirely expected. In fact, there will be no update of the threat or benefit associated with these cues or contexts. So if we apply this very in a schematic way to the fear memory system, that's how it would go. So there's a set of limbic brain regions, notably the amygdala, which is in charge of the fear memory, what we call the fear memory. And every now and then, some unexpected or slightly unexpected aversive event is going to take place, and this information will reach these regions together with sensory contextual cues. So when these are happening, the limbic brain regions, based on all these cues, are able to decide for a certain level of threat, for example, and they will propagate this threat information to executive structure, like the PAG, and these PAG will convert this threat into actual behavioral expression, right? So whenever some event occurs, an aversive event, what happens is that there's some comparison between the level of threat, the level of expectation, and the occurrence of this event. And if this event turned to be not entirely forecasted or properly forecasted, then what is called a fear prediction error signal is generated and fed into the memory update system and will trigger an update of memory. Okay, so then the fear learning is a lot about, predicting systems. So we would expect that the cerebellum might be playing a role into this. Okay, so we know for a long time that the cerebellum is protecting to the periapical gray. There's a long line of papers showing this, and we could easily reproduce these findings. We actually tested with optogenetics that this pathway was active, and when we stimulated it, we were able to trigger, to excite PAG neurons. It's mostly an excitatory pathway. Also, this pathway contacts both excitatory and inhibitory neurons. And the next question is, what happens if we manipulate this pathway when we perform fear conditioning? So again, we use the intersectional approach to express an inhibitory dread or excitatory dread only in the cerebellum PAG neurons, cerebellar neurons, the fastigial nucleus neurons actually, that projects to the periapical gray. And there we go. So what we do is either we do nothing, or we inhibit the cerebellum PAG neurons, or we excite them. So we increase by 50% or reduce by 50% their firing during the learning. So as you can see, the reaction to this repeated association of sound and shock is not changing much according to the cerebellum PAG pathway. Actually, that was slightly surprising, because the PAG is supposed to set the level of freezing, but in that case, that's not the compartment that the cerebellum seem to be acting on. We don't see much difference, but the difference comes the next day when we probe memory. And what we found is that if we had excited the cerebellum PAG during the association, it was pretty easy to extinguish those fear memories. And on the other hand, if we had inhibited this pathway during learning, the next day was harder to extinguish this learning. So somehow this cerebellum PAG pathway is performing a bidirectional control over fear learning. It either can prevent the association or reduce the strength of the association or reinforce it. So that feels very much like the cerebellum fiddling with the fear prediction error. So to test that, what Jimena Frontera performed is optogenetic experiments. So the experiments I just showed you use chemogenetic. That's pretty coarse, and that does not allow to, that's continuous, and that has very low temporal resolution. So what Shadmehr did was she excited the cerebellum PAG neurons only at the time where the shock was happening. It's really at the time, at the shock, that in fact, the brain has to make a decision whether the shock was expected or not expected. And if it was not expected, some memory update has to be performed. And of course, there are limitations to this approach because the optical fiber that we bring in is not going to cover the entire fastigial neurons. So we are not necessarily infecting all the fastigial PAG neurons with our strategy, and we're not necessarily exciting all of them. So we don't expect something as big as what we observe with the chemogenetic approach. But yet, what Shadmehr found was pretty much consistent in terms of direction with what she found with chemogenetic. So just exciting the cerebellum pathway at the time where some prediction error signal has to be constructed, just doing that at this time of conditioning is creating a memory which is slightly less easier to extinguish afterwards. Okay, so if we go back to the schematic, what this study showed is that in fact the cerebellum is also part of this circuit. We know from other studies that it receives from limbic regions and a lot from the sensory cues as well. And what we found with the chemogenetic manipulation is that this pathway participates to the memory update and very likely through a fiddling with a fear prediction error, which is now the signal we're trying, of course, to record to complement the study. Okay, so I'll try to, I'm slightly slow, sorry. I'm trying to reach this fear extinction learning, so the other type of learning that we probed in that. So fear extinction learning brings a new partner in the story, which is the prefrontal cortex. And just to summarize in a very simple way, what the prefrontal cortex seems to be doing in this fear extinction system is to make some decision about the maintenance or the suppression of the fear state and the update, of course, of the figuring that the cue can trigger a suppression of the fear state. And it does that by multiple projections, both to limbic regions, intercalated nuclei in the amygdala, and projections to the ventrolateral PAG, which is altering the processing. Okay, so we need to connect the cerebellum. If the cerebellum is going to do anything to the prefrontal cortex, we need to connect it. And it turns out that we knew from literature that there's a pathway that links the cerebellum and the prefrontal cortex by way of the mediodorsal thalamus. So if we look at cerebellar terminals in purple, there are some in the cerebellar nuclei. You can see them very clearly, I think, in the lateral part of the mediodorsal thalamus. And they come close to these green neurons that go to the medial prefrontal cortex. So what Shadmehr did also was performed transsynaptic labeling by viral approach using adenoviruses that jump one synapse. And this allowed to express the tomato reported fluorescent protein in the neurons that receive from the cerebellum in the mediodorsal thalamus. And what is interesting, I just make a zoom here to show you this. What is interesting is if we look at the terminals of these neurons that project to the prefrontal cortex, in fact, we find that the axon is all over the place, but we find a bunch of them in the layer 1 again. So layer 1 is again, we're falling in this multifocal sort of thalamus. Okay, so now chemogenetic manipulation of this. What Shadmehr did was here inhibition of the fastigial mediodorsal pathway during these repeated sound presentation. And what she found is that whenever there was this pathway was inhibited, animals that were expressing high level of fear, of threat, ended up really being stuck in this high freezing state, and they couldn't get away with it. There was almost uninterrupted boots of freezing during the whole duration of sounds. So it's really doing something with fear. Well, the mediodorsal is not the most classical source of regulation of freezing yet. Turning to the literature, there was this work from Lee and colleagues, which by a number of arguments, so this is just one of the arguments, show that the firing patterns in the mediodorsal thalamus were impacting how much fear expression and fear extinction was taking place in the animals that had undergone some acute fear conditioning. So particularly what they found is that enhanced expression of fear was found when there was a lot of bursting in the mediodorsal neurons. And indeed, we found by recording the mediodorsal thalamic neurons in our mice that they were expressing a number of bursts, and that the inhibition of the cerebellum-MD pathway was in fact enhancing the number of bursts that were observed there. So this is not necessarily very surprising, because we are removing an excitatory drive on the thalamic neurons, and we know that bursts are particularly easily generated in thalamic neurons if they start being excited from a very hyperpolarized state. So if we remove some excitation, of course, we'll get more bursts. So we felt it was worth, keep digging into this. And to do that, what we looked at, it was these patterns of activity in the cerebellum, which are the oscillations associated with freezing. So in these works from colleagues, you can see that there are very clear episodes of oscillation at the time of freezing. These are intense oscillation visible in the time frequency spectra on top here. And interestingly, these oscillations start before the animal really shows some freezing, and they stop a bit before the animal stop freezing. And indeed, they seem to play a causal role, because the rhythmic entrainment of the prefrontal cortex neuron is able to increase the expression of freezing. So these are not purely medial prefrontal cortex. They are also observed in the basolateral amygdala, and they seem to be entrained by the respiratory rhythm that is here observed in the olfactory bulb, and you can see is synchronous with the prefrontal cortex. Okay, so we were able to observe these oscillations in the medial prefrontal cortex. Actually, the power of these oscillations was increased when the fastigial mediodorsal pathway was inhibited. That's normal because we have more freezing. But we also found them in the mediodorsal thalamus. And interestingly, they're not the simple reflection of the prefrontal cortex. They kind of they come and go. And every now and then they show some nice synchrony or phase shift. And if we look at by generalized partial directed coherence, we can see that they're for the most part driven by the medial prefrontal cortex, and that's indeed further supported by the fact that if we look at the latency between the peaks of the oscillations in the cortex and the mediodorsal, we find that the cortex is slightly before the mediodorsal, a few milliseconds, which is compatible with the direct synaptic projections from the prefrontal cortex mediodorsal, which we know by other means that they're very powerful. Okay, so back to the burst. It turns out that the bursts are, in fact, phase-locked to the oscillations. This is not entirely surprising, because oscillations are alternative periods of excitation and inhibition, and we know during the period of inhibition that thalamic neurons are going to be more hyperpolarized, so that if they receive excitation, they will be more prone to produce a burst. And okay, that's the case. What's more surprising is, in fact, the fact that the bursts are in fact followed by higher oscillations in the prefrontal cortex. So there are some mechanism that links these bursts, or that make the fact that we have bursts and then later on higher oscillations. And in fact, those oscillations are reflected also in the mediodorsal, back to the mediodorsal, because if we look at the coherence, so this is time frequency coherence around burst. On 0, you have a burst for one thalamic neuron. So you can see that there are stronger coordinated oscillations in the mediodorsal and the prefrontal cortex. And this lasts for a couple, 1 or 2 cycles of the 4 Hertz oscillations after the burst. So overall, what this shows is that the mediodorsal thalamus is participating to the maintenance and suppression, mostly maintenance here, because whenever there's burst, this is amplifying the oscillations, and this will, of course, produce some feedback in the system. And so the mediodorsal is probably not doing this only with the prefrontal cortex. There's other regions which are not identified here, probably linked to the olfactory system, because that's how the olfactory bulb, the respiratory oscillations are fed in. And the cerebellum comes into it. And when we suppress the cerebellar input, we find that we have a lot more synchronous oscillation. So the physiological role of the cerebellum is to destabilize this fear expression state by acting on the mediodorsal thalamus. Okay, so running late in time. I'm putting this this slide because I think the old textbooks that I studied on were not making much of the thalamus and thalamic diversity. And this nice review of Francisco Klaska in this recent thalamus book really shows very nicely the diversity, the presence of multiple class of thalamocortical neurons. So the ventrolateral, the classical cerebello-thalamocortical pathway goes through that sort of thalamic nuclei. But I've showed you that there are other ones, and these seem to do different things. In the one to the motor thalamus is very much into closed loop organization that's suitable for internal models, anticipatory corrections, and so forth. Posterior medial does something quite different. That's a coordination of cortical activity. Mediodorsal is also about coordination here in the thalamocortical system, but probably also with other region, and that allows the cerebellum to control over control of this coordinated activity is really important for behavioral expression of a fear state. Okay, but that's only for the thalamus. What I showed you also is that the cerebellar projection to the midbrain is doing something that is happening in the forebrain, and that seems to be doing something with the prediction error type of activity. And so that's very likely not the end of the game, because amongst the midbrain structure that receive from the cerebellum, you find these dopaminergic regions that are known to encode and participate to brain function by encoding prediction error. So definitely I expect to see a lot more of these pathways in the guidance of behavior and learning in the brain. If we look at all these classes, in fact, we find that pretty much each category of thalamic neurons, in each category, there's at least one nucleus that receives substantial cerebellar inputs. And therefore, this should be reflected in different reactions of the brain into cerebellar manipulation and different sorts of cerebellar manipulation. So I think there's definitely many more to come, and I hope to see many young scientists join to understand what's going on there. Okay, so I'm done. Thank you all for your listening. So what I showed you is the work that we developed with Daniela Popa. So pretty much everything is work co-directed with Daniela. This is the place where we work in Paris, and we have a fantastic team. Always happy to have young scientists join us for a postdoc, and I thank you for your attention.
Okay, Clement, thank you so much. Let me start by asking you a question about the fear conditioning results that you showed and the effect that you showed on preventing extinction by inactivating, using dread, the cerebellar nucleus. Now, the cerebellar cortex presumably will continue to learn from the prediction errors. What happens the day after you remove the inhibition in the nucleus?
So we find that as soon as we remove this inhibition, actually the animals start dropping very rapidly their response. And for me, it's not entirely clear whether some degree of extinction has undergone underneath this very high expression of fear, or whether it's something that happens very rapidly in the system. So, but I think I'm not sure that in the case of extinction learning, I don't think that the cerebellum is learning this. I think the cerebellum is making an assessment that is propagated to the limbic regions to decide how much, in which state the brain should be. And of course, deciding on the state is going to have then an effect of learning. So it's kind of an indirect thing. My belief on that is. But as long as we haven't really identified what's happening with these bursts and oscillations in terms of cellular targets in the prefrontal cortex, I think we're a bit, that's more waving hands arguments at that stage.
That's great. Let's see if there's other questions for Clement. Yes, Violetta, go ahead.
Hi, Clement. So nice to see you, and thank you for the great
Hi.
talk. Yeah, I'm particularly interested in the second part of the talk on the emotional value of the cerebellum. And in my PhD that I'm finalizing now, I was able to see that there is a dopaminergic modulation of the FN, actually, of the fastigial nucleus. And we could see that actually the valence, so I was working with skill reaching, so it's a goal-directed action. But my question is, do you think if the deep cerebral nuclei have this role of modulator, like you showed and also Detlef showed, would you say that the DCN could also modulate the interaction of basal ganglia and the prefrontal cortex maybe, and then modulate the valence of activity? I don't know if you have any thoughts on any takes on this. It's just a very open question, but yeah, thank you.
Yeah, actually, I'll make it even more open. I think currently, I'd say if there's one aspect in this field that makes things very complicated, is that we don't know exactly how the collaterals of the neurons are really organized. We have so far very little knowledge, and we're still expecting the neurolite people, or the people able to reconstruct many cerebellar nuclei neurons. We're still waiting for them to provide for a database with numbers on these collaterals. So we know there are collaterals, and we know that whenever we have a circuit, we touch some neurons, we are going to affect multiple pathways at the same time. So in the case of the mediodorsal system, the mediodorsal experiments, what we did is we verified we had the same sort of effect if we just inhibited the cerebellar terminal in the mediodorsal. But if we go in general by manipulations of the cerebellar nuclei, in fact, we are going to have multiple effects, probably synergistic effect, and we're not dealing with one target structure, but in fact a circuit, and the cerebellum is part of a circuit and is shifting the state of the circuit. So to give a more focal response to your question, these limbic neurons, cerebellar neurons, some of them are sending collaterals to the dopaminergic system, and I would expect, but I think it's going to be a hell of a work to prove that. I would expect that, in fact, we get synergistic effects. So typically, I wouldn't be surprised that the effect we see in fear extinction is in parallel, maybe using the same types of neurons, performing some dopaminergic recruitment, which is known, for example, to perform a modulation in extinction, and is related to extinction learning. So I don't think that we should, of course, for carrying and for explaining things, I think I ended up always making things a little bit simpler than I believe they are. But definitely in the case of recruitment of the dopaminergic system, I think wherever we'll go in learning and valence modulations that involves the cerebellum, it's likely that we may have a combined effect through dopaminergic neurons and other regions as well, definitely.
Thank you so much. There are 3 other people that have raised their hands. So, Jen, do you want to go next?
Hi, Clement. Thank you for that.
Hi, Jennifer.
I was going to ask you to just speculate about, you motivated by talking about the cerebellum being good at doing predictions, and that's why it might get involved in the kinds of tasks that you were talking about. But I would kind of imagine that most of the brain is doing predictions. So what do you think is special about the kind of prediction that the cerebellum's capable of that gets it involved in like fear conditioning, say?
Yeah,
what kind of prediction is it doing that's different than the prediction that I think the rest of the brain does?
So very good point. So the 2 things it's very good at is associative, because the degree of heterogeneity of elements that can be associated in the cerebellum is very high. Maybe like in the hippocampus as well, which is also a big integrator of things that have little to do with another. It's almost hypothesis-free in terms of association. And that's quite different with many other regions which are a lot more dominant in what they treat. Of course, information is going all over the place, but in the case of the cerebellum, there's really a blender in the granular layer that is aimed at bringing in loads of things. And so I think for whatever the cerebellum, so whenever the cerebellum makes a decision into the degree of expectancy of a threat, for example, I think it has an ability to take into account a lot more things than, for example, the amygdala. Of course, this is if it's very simple, if it's a sound and the sound is going through the auditory thalamus and then directly to the amygdala, that's going to be easy. If it's a complex configuration that may even involve, I don't know, body orientation in space or something like that, well, I think the cerebellum has it. And it's very able to say, well, maybe the sound is something common, but we know that in that configuration, sound is relatively safe, where in that one, it's not so safe. So I think that's part of it. So that would be for, let's say, fear conditioning. In the case of fear extinction, I think it's, so then, okay, the idea is it's really a short-term assessment, because the other characteristic of cerebellum is that the temporal integration that can go on in the cerebellum is very short. You can't really rely only on the cerebellum to relate things that happened 10 minutes ago. You really need the rest of the brain to bring that in the short time window to make the cerebellum do the work. So the other, in the case of the fear expression during the sound after the pairing, I think it becomes, so it's pertaining to, again, this is going to be speculative and more about my intuition. But the idea there is that in terms of states, there are a need to make continuous choice. Should I should I go on, or should I move to something else? And that's true for being in the freezing state or non-freezing state. But of course, you can extend this to many other situations. And again, I think these decisions can be very subtle and require to integrate many more information. But what the cerebellum is going to do is not decide what you're going to do next morning. They're just going to say, okay, this is my assessment of what's going on in the last second or 500 millisecond. And based on that, I think I'm going to tip the balance in one direction. And that's where I would expect the cerebellum to operate the way I showed this, maybe in the emotional domain, but maybe also in other sorts of cognitive related functions. It's just making helping short-term decision. And that would be for the kind of things we're doing. I think there are other aspects linked to attention that obviously may go through these pathways, but we have very little data at that stage.
All right, let's move to Shogo. Shogo, you have a question?
Hi, Shogo. So many nice people.
Yes. Yeah, thank you very much. Thank you, Clement. This is really wonderful, story. And so I have two questions related to results question and also related to the previous question. And the first one is about the negative prediction error in fear extinction. So you showed the pathway of the cerebellar PAG pathway is contributing to the extinction, extinction process, and then how that this mechanism contributes to the negative prediction error, the mechanism. So do you think this pathway enhance the negative prediction error in the fear circuit?
Yeah.
So this is my first question. Sorry.
Yeah. Well, let me just answer it briefly. So actually, this pathway modulates fear extinction. And
Yes.
so we observed that, and the group of Richard Apps also did a very nice story confirming that. So yes, definitely, I think so fear extinction is a complex business. In fact, it's not a simple thing. I don't claim we exploited it all. I think we focused on what's happening that involves the mediodorsal, but I think I think all these things are circuit aspects, and the PAG contribution. Maybe I would expect through the dopaminergic neurons, because the PAG projects to the VTA as well. These pathways very likely also performing some prediction error computations that are pertaining to extinction, not simply learning or association.
I see. Okay. Thank you very much. And then my second question is about the time scale of the prediction, and also the time scale of the cerebral function. So you showed the cerebral contribution on the fear conditioning, and in the time scale of the 30 seconds or something like that, and it's a really big time scale, and you showed the fastigial nucleus is involved in that kind of time scale. And it's surprising to me because usually the fastigial nucleus is controlling saccharide eye movement or something like the motor control. So the time scale is a millisecond scale. So there's a huge gap from the millisecond scale and the 30-second scale. So how the cerebellum can contribute to that kind of large time scale with the prediction?
Yeah. So I don't know if Sasha is online, but she would say, of course, some neurons are very fast, but some are not. And she would be very clear about the fact that we have diversity in fastigia, but still, there's still a problem of time scale. Well, it turns out if you look at the activity of the PAG, and we've seen it, we have this in our data, but others have seen it. What happens is that you have a very clear response. So when you've performed the association, you have a very clear response at the onset of the CS. And my understanding of that is that the level of threat is set at the onset of the system. So so what happens? So I don't think that the cerebellum is performing the association, right? But the cerebellum is. So the moment you start playing a sound, as soon as you started associating the sound with the shock, the brain is shifting to a different state and is making a threat assessment, which may not be very temporally structured. I guess depending on the detailed parameters, the animal may gain the ability to decide and the amount of training. But I think with the 5 set, they don't know that at 30 seconds, the shock, it's exactly the time. So they just have a global expectation why the sound is going on for that sort of training. Of course, parameters will matter there. So where the cerebellum, so there are 2 aspects of it. So whenever you start playing the sound, you have a first shift, and you will get a prediction error at that time, and the cerebellum might be also operating at this moment. And the moment you have the shock that comes in, then you have, again, a correspondence between the identified level of threat and the intensity of the event, right? So I think where the cerebellum is expected to play a role at these moments, the moment you start setting, okay, we have to update the threat level, and the moment that something happens, how does that compare to the threat level? If we go for the detailed correspondence. And of course, there's a global threat, and there's, as I was saying earlier, there's instantaneous elements that can be integrated into that computation and refined. And I think that's where the cerebellum would, my guess is that that's where the cerebellum is going to be the main player.
All right, let's go to the last question.
Thank you very much.
June, go ahead and ask your question.
Okay, so my question is, thank you for the talk. I read your paper, now I think I have better understanding. So my question is, do you see any difference in the activity in the DCN after learning or after extinction? Because you're doing in vivo recording. And my second question is, what is your thought about any connection between the cerebellum and the amygdala?
So we haven't been able to see simple shifts of activity, like the fastigial neuron starts firing a lot more, things like that. But it may definitely happen. But these are difficult to disambiguate from changes in behavior. So where we see differences is more if we start looking at coordination between fastigial and PAG neurons, for example. And there we see changes associated with the degree of coordination between the fastigial neuron and the PAG after learning. We've had evidence that this is unpublished, but we've seen that. I don't know how robust this is, but we've seen it. That is definitely these neurons tend to be more better coordinated, particularly in relation with the occurrence of a sound. For the pathway to the amygdala, actually, it's interesting you asked, because when Jimena Frontera started doing all these experiments to map the circuitry, basically, and really look at the detailed connections where they were found, the primary target for us was the cerebellum amygdala pathway, which we found through the parafascicular thalamic nucleus. And so we believed, okay, this is it. And I think that's amongst the first manipulation, that chemogenetic manipulation that Jimena performed. And she didn't see much of an effect. So there was some effect, indications of an effect in the anxiety. So it seemed to be more related to a non-specific threat level, which was not what we expected or would have guessed. This pathway has been better characterized, of course, since then. But still, we don't see a big effect in fear learning by this pathway. So the short answer, there's definitely a pathway, but what it does for fear learning is in our hands is absolutely not clear. So our take is that it's doing something probably different from all the aspects, all the dimensions we've been discussing there. There was your first question was the second, sorry.
The first question, I think you partially answered it, is whether after learning and the extinction, change.
So that was it.
You see some kind of increase between the, no, you do.
No.
After learning, do you see any increase or decrease?
Yeah, so nothing simple that I would trust, I think, because of course the behavioral state is going to be reflected definitely on if you just measure the speed of movement and so forth, this will show up as well. So because the behavior is different, then the vestigial state is different. You can't make it causal at that stage.
Thank you so much, June. Clement, we are out of time, and thank you so much for waiting around and answering all the questions.
Okay, my pleasure.
See you all next week.
Okay, bye, thanks.